Hello everyone and welcome back to this online NPTEL course on aquatic geochemistry. We had been discussing about ocean chemistry in our previous lecture, and in today's lecture again we are going to continue discussing some more. So, we stopped in the part where we have seen oceanic current formation and the Ekman spiral formation, and this is the circulation of the ocean, and we have mostly two kinds of circulation. First is the ocean current, okay, that is there, and secondly is the depth circulation. Okay. So, there is two types of circulation. One is this spatial circulation which is reflected as oceanic currents, and that is the mixing of the entire global ocean, and also at every location we can see that there is some depth circulation also happening. Therefore, when we talk about composition, the spatial variations of dissolved element concentrations, they reflect this ocean circulation. If you remember, when we were talking about sampling, we were saying that the concentration always is hinting us that how the ocean current is moving. If these are surface samples, then it will reflect how the ocean currents are moving from one place to the other. In fact, the chemistry is extremely strong evidence that the current is moving. Okay. So, these kind of ocean circulation which is driven due to the difference in solar heating or insolation, the unequal heating is operating, and also because of deep circulation, the depth circulation, there are distinct mechanisms that is happening, and for every element, not only the group of elements that we can talk about. So, there is the major anions, the major cations, then there is some trace elements, there are some minor minor elements. So, we call them minor because it is not even major, it is not even trace, they are minor nutrients and concentrations. So, there is a major, there is a minor and the trace. Now, when I talk about trace, the trace are really extremely low. So, there can be ultra low concentrations of particular elements, and even if you in fresh water we know iron, it can be in few hundreds of ppb, and in the ppb level. In ocean, that concentration is extremely low. It can be in ppt levels also. Ppt means parts per trillion. It is not parts per thousand, but now parts per trillion level of composition. And also important to say that the sea water composition, the major ion composition is generally very well fixed, and we are going to look into that in just a minute. So, this is the way the currents are formed in the ocean, that is due to the Ekman spiral and due to atmospheric circulation. As you can see, in the northern hemisphere, the oceanic currents move in a clockwise direction. Okay, so, this is the clockwise direction that is moving, and in the southern hemisphere it is the anti-clockwise motion how it is moving. So, the every ocean, so the Atlantic Ocean, the Indian Ocean, the Pacific Ocean, the currents are moving. It is along the coast the currents are moving, and in the equator we have the equatorial currents separated by the equatorial counter current. In the polar region also we have the Antarctic circumpolar current, and in the northern pole also we have this Greenland current and Norwegian current and the Labrador current. So, all the currents are are divided in the different regions and also according to the different oceans. Now, what does this lead us to? So, we understood the surface circulation, the surface circulation. Now, let us look into the deep circulation. Okay, the deep ocean circulation is driven by the density differences. Okay. So, the surface ocean circulation is driven by the wind currents and the unequal heating of the unequal temperature, that is there. But in the deep ocean, circulation is driven mostly by the density, the heavy and light water, how it is mixing, which again is controlled by temperature and salinity, and therefore collectively called thermohaline circulation. Okay, it is controlled by the thermohaline circulation. Now, the ocean is very stably stratified. Okay, there are very fixed blocks that we have seen, with the vertical water moving mainly through upwelling and downwelling zones. Okay, so, there are regions where the winds or the currents cause divergence of the water. So, if the waters are getting diverged, if the surface, if the water currents and if the winds are getting diverged, what is happening? This place is having less mass. So, what happens is, from the depth of the ocean, the water will rise up to fill this place, because the surface water is moving away, the surface water is moving away. So, the deep water has to rise up to maintain the maintain the level. Again, when there is downwelling, when the ocean currents are meeting, when the winds are meeting, at those areas there is more water on the top surface, on the surface. So, that creates a pressure and the water moves down. Okay, so, this is called downwelling, where due to the convergence of currents, due to the convergence of currents, the the water pressure becomes high on the surface and the water moves down. Okay. So, due to the movement of different currents, this is created. Beneath the surface mixed layer lies the thermohaline, where temperature decreases rapidly with depth. So, the first few meters of the ocean surface is called the mixed layer, the surface mixed layer. Okay. So, for everything this is called the surface mixed layer. Okay. The water is continuously well mixed there. So, beneath this surface mixed layer is another layer. Okay. This is another layer which is called the thermohaline, and here the temperature decreases rapidly with depth. So, in this zone the temperature is rapidly decreasing, as you can see from the surface it is rapidly decreasing. After this particular depth it becomes very constant, it is decreasing, but the decrease is the gradient is very low now. Okay. Here the gradient was very high, it was very rapidly decreasing. Here it is almost like a constant line, and it is not decreasing at a very fast rate. So, this particular layer where it is rapidly decreasing is called the thermocline. Same thing where the salinity is rapidly changing, where the salinity is rapidly declining, that layer is called the halocline. Okay. Then just because density is related to salinity and temperature, the layer where density therefore is also rapidly decreasing is also called the pycnocline. Okay. So, together these gradients are creating a strong barrier. Okay. So, a strong barrier is created, and that separates the well mixed surface layer from the deep ocean. Okay. So, this is the deep ocean and this is the surface layer, okay. The surface well mixed layer, and this particular thermohaline pycnocline layer, this layer it is like a boundary, it does not allows a lot of mixing happening. Only what can happen is a lot of settling down, like particles settling down, that kind of this thing is happening. Okay. And where there is mixing, that time there can be a break, because just because there is a mixing going on. Mixing means in the upwelling and the downwelling regions, or if there is in the polar region, if there is polar region, the water column may be nearly isothermal. So, the mixing the thermocline, the halocline, the pycnocline, they can be little different. So, what results is The chemical implication of these depth mixing is that we get two separate boxes. So, we have the surface ocean and we have the deep ocean, okay. And it becomes very useful to us because it becomes like a two box model, and it is very useful for all the chemical interactions, because this is separate, the top model, the top box is separate, the bottom box is again separate. The surface ocean, what does it influence by? It gets influenced by the exchange of gases with the atmosphere, okay. It receives all the riverine inputs. It hosts nearly all the photosynthetic activity, as light penetration is limited to 100 meters, right. So, light itself beyond 100 meters, there is no light, it is very dark. Divers who go in, you must have seen that they will have these lamps, lights attached to their helmets. So, those kind of gears they are wearing. So, it is very dark inside the deep ocean. So, light does not enter to, there is the twilight zone. Beyond the twilight zone, the ocean is absolutely dark, okay. So, photosynthetic activity is very much limited only to the top 100 meter, and therefore, a lot of life related activities is limited to the surface ocean, okay. A lot of animals are there, a lot of creatures are there. Only animals who are who can go down, deep diving animals, they can only go down, and the life cycles and the processes that the larger animals they have is very different from these animals that is living in the surface ocean. The deep ocean, they act as a primary sink for sinking organic and inorganic particles, okay. They represent the main pathway for long term removal of dissolved and particulate material from the ocean. So, the deep ocean is very important if you are looking for long term changes, okay. If you are looking for global elemental cycles, exchange between the two boxes occurs via advection if there is any upwelling or downwelling, or due to particle settling. So, just the sinking of particles, either organic matter or even inorganic sediments, if it is close to the estuary and deltas, then there are a lot of inorganic sediments that is also getting coming in, and also the the orthogenesis that is going on, the clay formation that is going on, the carbonate formation that is going on, the evaporite formation that is going on, all that kind of particles they can also start settling in, okay, they can form. And you need to know that the ocean is not having a simple this kind of topography. So, there are many depth breaks, okay. So, this like the continental shelf is there, the continental slope is there, the deeper part of the ocean is actually very narrow parts, okay. So, it is not that the entire ocean has the same depth. So, when we are talking about coastal regions, the depth of the coastal region is very different. In the shelf area, the kind of topography is again very different. So, due to this, vertical mixing is different, formation of different kinds of sediments where they are settling, that is also different, and all these affects the chemical composition. And the crossing of the pycnocline is restricted and is only occurring in few regions, and those are the zones of intermediate water formation. That means meeting of two different currents and intermediate water formation is happening, or there is a deep water formation like the downwelling is happening and all that, where surface waters acquire characteristic temperature and salinity properties before sinking into the ocean water. So, these are again a different physical circulation happening itself because of changes in the ocean water properties. So, this again is like very specific and very unique in particular areas. So, let us see what is the sea water made up of. Major ions, the 6 major ions it makes up 99.8% of all the dissolved solids in the water. See, if you have 1 kg of water, okay, then 965.6 gram is water, and salinity, that is salts, total salts, is just 34.4 grams. So, in 1 kg you have 34.4 grams of salt. In that 34.4 grams of salt, 18.98 grams is just chloride, only chloride. Then we have sodium, that is around 10.556 gram. Then we have sulphate, then we have magnesium, bicarbonate, calcium, potassium, and everything else is just this much. So, this is the minor and the trace elements. Okay. So, this is extremely low. So, just imagine more than half of the total dissolved solids is just chloride, and the other big chunk is sodium. So, sodium, chloride, sulphate, magnesium, calcium, bicarbonate and potassium, these are the major ions that is going to make most of the seawater, and everything else, silica, nutrients, silica is also a nutrient. So, silica, nitrates, phosphates, all the trace elements, their concentration is going to be very, very low. In fact, some of the trace elements, the difficulty it becomes is that of their extreme low concentration, it is very difficult to measure them with accuracy in the even with our good technologies. So, there are very few labs that does work with ocean samples and is trusted. Also, the kind of composition is also related to the the process that is happening. So, at certain locations where there is a lot of evaporation going on. So, if close to the equator, continuously evaporation, precipitation, the cycle is going on. Okay. Close to the mouth of the river, a lot of fresh water inputs coming, a lot of sediments coming, a lot of changes, exchanges are happening. Different kind of composition is there, where the lot of fresh water is getting frozen to form the ice. Okay. There again a lot of different processes are happening. So, this water cycle also increase, decrease of salinity, increase, decrease of water itself causes changes in its composition. But regardless of how the salinity varies from place to place, the ratios of the major ions in water, they are nearly constant in open surface ocean. Okay. It is the ratio is always constant. So, everywhere in the open ocean, the chloride is to sodium ratio is around 1.796, sulphate is to sodium ratio is 3.972. Okay. Except for where near the coastal regions, where there is or where there is the mouth of a river, that kind of place. So, this ratio is always maintained, and the oceans are well mixed. We say that their oceans are well mixed with respect to the major dissolved species. That means that their residence time in the ocean is very large as compared to the mixing time. We have discussed this when we were discussing about strontium and strontium isotopes in the marine record, and we say that strontium has a very long residence time and it is very well mixed. So, this is now again coming into picture because we are discussing about ocean and the elements, and residence time becomes a very, very important factor, right. So, the residence time ranges can sometimes range less than 50 years. For instance, for iron it is less than 50 years, and it can vary up to more than 50 million years for in terms of sodium, chloride. These have a very, very long residence time in the ocean. So, if the residence time is very low, if it is lower than the mixing time. What is the mixing time of the ocean? The mixing time of the ocean is around 1000 to 2000 years. Okay. So, if the mixing of the ocean is, if the residence time is less than the mixing time, then that element does not gets accumulated into the system anymore. It gets again recycled back onto the terrestrial system and it gets removed from the system. So, if the residence time is low, the accumulation gets low, the concentration in sea water is very low. If the residence time is very high as compared to the mixing time of water, then its concentration slowly becomes uniform. There is no particular event that will cause change in its concentration, it just gets well mixed. Okay. And it is specifically important to note that all the major ions, they have their residence times more than 1,000,000 years, which is many times much higher as compared to the mixing of the oceans, that is around 1000 to 2000 years. So, in general what we see is that the higher concentration elements, those that are existing in the higher concentrations, they have higher residence times. For instance, sodium chloride, calcium, magnesium sulfate, potassium, they have very high residence times as compared to other elements, for instance iron, manganese, any other trace element, they have very low residence times. And certain elements we take them as minor elements and not trace elements for seawater. For instance, strontium has a very good has a pretty good concentration. Lithium has a good concentration, carbon is as a minor element. So, there is a difference in the way that we report fresh water and ocean water, and you can see here the residence time is given in the x axis and the sea water concentration is given in the y axis, and this is again grouped in terms like which is which particular elements. It all depends on how the elements are getting in and going out. So, this balance decides how which element is going to stay for a longer duration, which element is going to get removed. So, the elements with longer residence time, they have a long time to accumulate in the ocean. Okay, they have a long time to accumulate in the ocean, and they undergo a large number of mixing cycles. Okay, because if you see here sulfate has 10,000,000 years. Okay. Now, if you consider that 10,000 years is the mixing time, then you imagine, so 10,000,000 divided by 1000 years. So, how many times sulfate is getting, so 1000 times it is getting recycled. So, that is how you can see that how many cycles it can undergo, and therefore, they are very well mixed. Okay. The elements which have short residence times, they are removed very quickly, and therefore, they do not get a build up time, right. Their concentrations do not get build up, and they exist in the ocean for less than one mixing cycle. So, for instance, like iron, which has a residence time of 50 years. So, this is much less than 1000 years of well mixing. So, in 50 years the iron is getting recycled. So, it is not getting even one mixing cycle. Okay. So, therefore, it is not getting enough time to get built up into the ocean. And what is it that is actually controlling the residence time? It is not the input rate, it is mainly the rate of removal. So, the rate of removal is actually determined by what chemical processes, what biological processes are happening, and that is actually deciding the composition in the seawater. So, if it is very biologically active, if it is chemically very active. So, for instance, iron, why is the residence time of iron so low? In fresh water, in the fresh water, the iron composition is not as low. Why is that the iron composition is so low in the sea water? That is because iron is a very important micronutrient. Okay, it is an extremely important micronutrient and it is utilized by the bacteria, by the microorganisms, okay, for their photosynthetic and the metabolic activities. So, iron is a micronutrient and is the limiting nutrient in the ocean. So, whenever there is a large iron coming in, there is a algal bloom and everything happening, and there will be it completely, the organisms they will take away all that iron and it gets just converted into organic matter. So, it is these biological processes that is taking up. Similarly, here is a process shown for manganese. Okay. So, the riverine manganese is coming, it is getting into the water, the ocean water. There is some near shear diffusive removal processes. Then there is dissolved manganese that is going on, and the dissolved and the particulate manganese, they are continuously exchanging, okay, between the organic and the inorganic phases, they are continuously exchanging. There is some particulate manganese sedimentation. Okay. Then again with the biogenic silica, with the organics, with the calcium carbonate, certain proportion of that manganese is getting settling down, it is getting removed, it is getting removed from the system. Okay. Again the diffusion of these pelagic sediments, okay, of these organics, biogenic, calcium carbonate, they can also supply certain a certain proportion of manganese that got settled, a certain amount is returned back. The hydrothermal vents, they are also giving out the manganese, okay. They are again getting coming as precipitation, they can again get precipitated out, they can form nodules. So, this manganese is getting removed from the water, from the dissolved it is getting removed and it is getting down as sediments, okay, as nodules, as inorganic sediments, as organic sediments, it is just getting removed from the system. So, such removal processes is what controls the concentration in the sea water. And elemental concentration was not very well known until the 1950s and 60s, and we realized that a lot of joint missions had to be done, because it was not the scientific community slowly understood that it is not possible for a single country or for a single team or a research team to do very conclusive research because of the vastness of the oceans. So, what they started is they started having multiple missions to do the physical and chemical oceanography. So, there were joint missions done by a lot of institutes all over the globe, okay, not only particular regions, all over the globes there were specific groups created and we had missions with different research objectives. So, we started the very early objectives was understanding the ocean circulation. Okay. At that time, understanding the ocean features, what is the depth, what is the what is the bathymetry, what physical features we are seeing now, we came to know through those observations and experiments. Similarly, there was a joint, it was preceded with the joint global oceanic flux study. So, that time there were again many objectives. The chemistry came mainly with the geochemical ocean section study, where they looked into particular sections of the ocean. Okay. They took up certain sections of the ocean and then they used to study those. So, geosex started. Then since the 2000s we started having the geotraces, which focused on the trace elements, because until then the trace element compositions were extremely difficult to measure, and the problem was mostly analytical, the technology was not advanced till then. So, with the advancement of technology, with the advancement of our analytical techniques, we started taking more precautions. So for instance, iron is in very low level, and what used to happen is these ships were built with iron. Okay, so they are metallic alloys. So when they go in the ships, then there is a lot of contamination from the vessel itself. The ship itself is like a contamination. Like if you are going in a ship, you are carrying things in the ship. So when we work in the lab, we work in a metal free lab. Like for inorganic people who are working in metals, we work in a place, in an environment where the air is clean of the metals. So that was a very big challenge when doing experiments or collecting samples in the sea, because these ships, they are already having a lot of contamination and background contamination, and in the sea water the level is so low that often the blanks used to have higher values as compared to the sample itself. So it became a real challenge at that time. With the geotraces coming in with more advanced analytical procedures, basically with the advancement of technology, we now are able to analyze many elements, and at extremely low levels, ultra trace level of measurements, and for each and every element the kind of composition we see is very different. Okay, and that is due to the different biological and physiochemical processes that is happening. For instance, here is an example of phosphate and silicate, and the differences that are there in the North Pacific Ocean and the North Atlantic Ocean. Okay, here also there is the North Pacific Ocean and the North Atlantic Ocean. Now the phosphate is the line, and cadmium is represented by the symbols. Here also silicate is represented in the lines, and zinc is represented as the symbols. Okay. So when they measured, their point of checking, like we have to check like whether there are errors or not. So first of all, there has to be coherence in the data set. So if this data point lies somewhere here, you know that this is a wrong data. So there has to be spatial and depth coherence, and second of all, there also has to be correlation between the geochemically similar elements. So if cadmium and phosphate, they are correlated, then you know okay, your data is very good. If silicate and zinc, okay, micronutrients, so they are correlated, so you know okay, the data is correlating well with its similar element, then the data is good. And here it shows the different kinds of signatures, that is because of the different processes that is happening. So phosphate concentration increases more rapidly, it increases rapidly with depth as compared to dissolved silica, and that shows the regeneration mechanisms, the difference. So phosphorus gets released during the degradation of soft tissues, the organic matter. So it gets released into the water, okay, while silica is stored in the skeletal material, the test that they call. So the opal test, okay, that is the inorganic matter. So it is a very slow process, its dissolution of that skeletal material is a very slow process. Also the passage of the siliceous test through the digestive system of higher organisms, they can sometimes accelerate the silica dissolution. So how the larger fishes and larger organisms, how they are interacting and how they are digesting and how they are then releasing the silica, that is also controlling the silica dissolution. Elements that get rapidly regenerated during the organic matter breakdown, they are called labile elements. For instance, phosphorus, okay, these phosphorus, they are called so they are labile. So nitrate, manganese, cadmium, nickel, okay, they are rapidly recycled, Okay, they are the more labile elements as compared to silica, which is more refractory, which is inside the skeleton and whose process is much more slow as compared to the other labile elements. So, that it is called the refractory element. So, every element, its behavior is different. So, when we look into ocean composition, our major cations and major anion compositions is fixed. So, this is the major cation and anion composition. Okay. So, this is fixed, the ratios are fixed, but for the trace elements and for the minor elements, there can be a lot of variability depending on the physiochemical processes. Okay. So, with this I am going to stop in today's lecture and I am going to continue discussing about trace elemental chemistry in the next lecture. Thank you.